The Musculoskeletal Knowledge Portal, or MSKP, is the newest portal in the Knowledge Portal network. Produced by a collaboration between the International Federation of Musculoskeletal Research Societies and the Knowledge Portal team at the Broad Institute, the MSKP aggregates, analyzes, and displays large data sets relevant to musculoskeletal traits and diseases. The MSKP is powered by the Human Genetics Amplifier data and software platform that was developed at the Broad Institute with support from the Accelerating Medicines Partnership in Type 2 Diabetes. At present, the MSKP includes several genetic association datasets for musculoskeletal phenotypes, along with epigenomic data from osteoblasts and other tissues. In the future, it will be expanded to include more data types, including transcriptomics, chromatin conformation, EQTLs, proteomics, and more. You can see a list of the datasets currently available in the MSKP on the Datasets page. Each section expands to provide details about the dataset. The MSKP has three major entry points to these results. If you have a gene, genomic region, or variant of interest, you can find more information by entering a gene name, chromosomal coordinates, or variant ID into the search box on the home page. Searching with a gene name or coordinates of a region takes you to a gene or region page, respectively, that summarizes what is known about genetic associations and epigenomic marks within that region. The top of this page has a traffic light symbol that shows you, at a glance, whether there are strong associations in the region, a green light, suggestive less strong associations, a yellow light, or no associations, a red light, within the datasets in the portal. Below the traffic light, the Phenotypes with Signals section shows the phenotypes for which there are significant associations. Green color coding indicates highest significance, while yellow indicates moderate significance. Clicking on a phenotype changes the display below that point on the page, on each of the four tabs, so that all of the results pertain to that phenotype. The top Variants tab on the Gene page displays a table of the most significantly associated variants across the region for the phenotype that you have chosen. The rows of the table are also color-coded by significance. Below the table, a locus zoom plot displays the associations across the region and two annotation tracks showing the locations of attack-seek peaks and DNase hypersensitive regions in osteoblasts. You can add more locus zoom plots for different phenotypes and datasets using the menus above the plot. You can also select tissues to display chromatin state annotations for those tissues. We'll cover the three other tabs of the Gene page in a future tutorial. Searching with a variant ID on the home page, or clicking on an ID anywhere in the MSKP, takes you to a variant page. The top section of the page, Associations at a Glance, summarizes the associations of that variant across all of the datasets and phenotypes in the portal. You can choose to display the best associations from individual datasets, or bottom-line p-values calculated by overlap-aware meta-analysis across all datasets. You can also view phenotypic associations across a large set of phenotypes from the UK Biobank. There's lots more on the variant page, but we'll cover that in a future video. The second major entry point to the portals is the interactive Manhattan plot. To see it, click the Explore by Phenotype tab on the home page and enter a phenotype. This tool lets you browse associations across the whole genome for any phenotype and dataset. Points are linked to variant pages, and the top associations are listed in a table below the plot, with links to gene pages for the nearest genes. The third major way to explore the results in the MSKP is to use the Variant Finder tool, which can be accessed via the Tools pull-down menu. You can specify multiple search criteria and retrieve the set of variants that answer all of those criteria. For example, variants that are associated with both estimated bone mineral density and height. Finally, under the Information menu, you'll find information about the project, help documentation, how to collaborate, and more. please contact us using the form at the help link with your questions and suggestions.